IB Biology, Topic 3.2 Chromosomes Point 9. A karyogram shows the chromosomes of an organism in homologous pairs of decreasing length. Before we begin, you must know that chromosomes are more clear or more visible during certain stages of a cell's life. They're visible in mitosis, but metaphase gives us the clearest view. So metaphase is the clearest view of the chromosomes. To better see these chromosomes, scientists use stains, and they're used specifically, so stains can be used to give a distinctive banding pattern. If stained at the right time and in the right way, chromosomes are quite easy to see and observe under a microscope. The best time to stain and observe these chromosomes is when the cell is dividing. And the process looks like this. So first you stain a dividing cell. Next, you burst the cell. So you burst the cell by pressing the slide glass. And the final step is to spread the cell around to see the chromosomes. To better visualize this, here you have an attempt at drawing a microscope. And when you look at something under a microscope, you put the thing on a sheet of glass, or a slide of glass. You then put a second slide of glass on top, and you squeeze them together. So in this case, if you take a slide of glass and put a dividing cell on it, put another slide of glass on top of it, smush and spread around, you'll be able to see the chromosomes, as long as you've stained it first and are doing it at the right time, so during mitosis, or even better, during metaphase. Once you've done all of this, you take a micrograph of the same chromosome. So this process is so that you can, or rather so that you're able to, so this allows you to take a micrograph of the stained chromosomes. What you then do with this is that you arrange them. So next we wrote that cells can be arranged. In this case it's digitally, although this used to be manually done. And these are arranged digitally by size and structure. One thing that we look for is the position of the centromere. As well as the pattern of banding. Because each chromosome, if stained correctly, will have a different pattern. And these 2 points make it easy to distinguish chromosomes of different types even if they are the same size. And if you don't remember, the centromere is this little thing in between the two chromatids that connect them together. So if you have a chromosome, this entire thing, these two sides are chromatids, and this little point in the center is the centromere. So depending on the position of the centromere, the pattern that the same chromosome has, it's very easy to distinguish the different types. So you'll be able to distinguish between chromosome 1, 2, 3, and so on, even if the chromosomes that you're looking at are the exact same size. Most of the cells that you'd take to look at under a microscope in this way will be diploid. So most cells are diploid. And this means that inside of the cell, you will see two sets of the chromosomes. So most chromosomes you'll find in homologous pairs. This makes it even simpler when identifying the chromosomes because they'll be next to each other. And then you take this and you arrange from largest, or rather the longest, to the smallest, or to shortest. And when you do this process, for all of the chromosomes that you're looking at, you'll end up with a karyogram. And what this looks like is you'll be arranging the chromatids next to each other, so the two sides of the chromosome, and you'll have chromosome number 1, number 2, number 3, 4, and all the way until you get to the sex chromosome. And depending on the cell you're looking at, that will either be an X or a Y. So you'll either have two long chromatids, or a long chromatid and a short chromatid, resembling the Y.